Science Quest, Unlocking the Universe Episode 1, The CRISPR Revolution and Gene Editing. Ever wish you could rewrite the code of life? Sounds like science fiction, right? Warm. Today, we're diving headfirst into the incredible world of CRISPR-Cas9, a gene editing tool so powerful it's rewriting the future of medicine, agriculture, and even conservation. Imagine your DNA is a vast, intricate instruction manual for building and running your body. Sometimes, there are errors in this manual, typos that lead to genetic diseases like cystic fibrosis or sickle cell anemia. CRISPR is like a highly advanced find and replace function for this manual. It allows scientists to precisely target and edit specific genes, correcting errors or even introducing new ones. At its core, CRISPR is a naturally occurring system found in bacteria. These tiny organisms use it as a defense mechanism against viruses. They essentially remember viral attacks and cut up the virus's DNA upon reinfection. Scientists cleverly harness this natural system, create a tool that can be programmed to target any DNA sequence we choose. Let's break it down. CRISPR-Cas9 consists of two main components, Cas9, an enzyme that acts like molecular scissors, and a guide RNA molecule. This guide RNA is like a GPS guiding the Cas9 enzyme to the precise location on the DNA where the edit needs to be made. Once there, Cas9 snips the DNA, allowing scientists to either disable a faulty gene or insert in a new, corrected one. Think of it like editing a document. You use the find function guide RNA to locate a specific word target gene, then use the replace function Cas9 to correct or change it. This incredible precision is what makes CRISPR so revolutionary. The potential applications of CRISPR are staggering. In medicine, it holds immense promise for treating genetic diseases. Clinical trials are underway for conditions like sickle cell anemia and beta thalassemia, with early results showing remarkable success. Imagine a future where these devastating diseases are simply cured through a gene edit. Beyond human health, CRISPR is transforming agriculture. Scientists are using it to develop crops that are more resistant to pests, diseases, and harsh environmental conditions, potentially addressing food security issues globally. We're even seeing its use in conservation efforts. Imagine restoring endangered species by correcting genetic defects. This incredible power comes with significant ethical considerations. The ability to edit the human germline, the genes passed down to future generations, raises complex questions about unintended consequences and potential misuse. Strict regulations and ethical guidelines are crucial to ensure responsible development and application of this technology. The future of CRISPR is bright, with ongoing research exploring its applications in areas like cancer immunotherapy and the development of novel gene therapies. Scientists are constantly improving the precision and efficiency of CRISPR, making it an even more powerful tool for addressing some of humanity's most pressing challenges. Let's test your knowledge. What are the two main components of the CRISPR-Cas9 system? A Cas9 and guide RNA, bDNA polymerase and ligase, C restriction enzymes and plasmids. Pause for audience response /poll. The correct answer is A. From curing diseases to revolutionizing agriculture, CRISPR-Cas9 is transforming our world in profound ways. Next time on Science Quest, we'll explore another groundbreaking technology, the quest for fusion energy, a potential solution to our planet's energy needs. Join us then as we unravel the mysteries of the stars and the power they hold. CRISPR gene editing, CRISPR-Cas9, gene therapy, genetic diseases, agriculture, conservation, ethical considerations, future of CRISPR, fusion energy, AI explained. Science Quest, Unlocking the Secrets of the Universe Episode 1, The CRISPR Revolution. Did you know that scientists can now edit our genetic code with the precision of a word processor? We're not talking science fiction. This is the incredible reality of CRISPR-Cas9 gene editing, a technology that's rewriting the rules of biology and holds the key to treating diseases, enhancing crops, and even reshaping the future of life itself. Welcome to Science Quest, where we explore the cutting edge of scientific discovery. CRISPR-Cas9 is a revolutionary gene editing tool derived from a natural defense mechanism found in bacteria. Imagine a bacterial immune system that remembers and targets viral invaders with pinpoint accuracy. CRISPR, short for clustered regularly into space short palindromic repeats, acts like a molecular GPS, guiding the Cas9 enzyme pair of molecular scissors to a specific location on the DNA strand. Once the Cas9 enzyme reaches the target location, it makes a precise cut in the DNA. This cut can be used to disable a faulty gene, insert a new gene, or even correct a single nucleotide, the building blocks of our genetic code. Think of it as a powerful find and replace function for our genes. The implications of CRISPR are breathtaking. Imagine a future where genetic diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease are eradicated. CRISPR offers the possibility of directly correcting the faulty genes responsible for these devastating conditions. Clinical trials are already underway, testing CRISPR effectiveness in treating various cancers, blood disorders, and inherited diseases. While challenges remain, early results are promising, offering a glimmer of hope for millions affected by these debilitating illnesses. Beyond disease treatment, CRISPR holds immense potential in other fields. We can engineer crops resistant to pests and diseases, increasing food production and reducing our reliance on pesticides. We could even develop disease-resistant livestock, improving food security and animal welfare. With such powerful technology comes significant ethical responsibility. The ability to alter the human germ and the genes passed down to future generations raises profound ethical questions. What are the potential unforeseen consequences of modifying our genetic heritage? How do we prevent misuse of this technology? These are crucial conversations we need to have as a society. Regulation and careful oversight are paramount to ensure responsible development and application of CRISPR. Transparency and public engagement are essential to navigate these complex ethical dilemmas. The future of CRISPR is bright, but it's not without its challenges. Scientists are continually improving the accuracy and efficiency of CRISPR, aiming to minimize off-target effects, unintended edits in the genome. Advances in delivery methods are also crucial, ensuring the gene editing tool reaches its intended target efficiently and safely. Imagine a world where we can prevent inherited diseases before they even manifest. Imagine being able to enhance our own immune systems to fight off infections more effectively. CRISPR holds the key to unlocking these possibilities and many more. What are your thoughts on the ethical implications of CRISPR gene editing? Share your perspectives using ScienceQuest CRISPR on social media. CRISPR-Cas9 gene editing is a groundbreaking technology with the potential to revolutionize healthcare, agriculture, and beyond. While challenges remain, the future looks bright for this powerful tool that's rewriting the very code of life. Join us next time on Science Quest as we delve into the mysteries of dark matter and dark energy, the unseen forces shaping our universe. Science Quest, Episode 1: Unlocking the Universe, Fusion Energy and the Quest for Clean Power. Did you know that the Sun, that giant ball of fire 93 million miles away, is essentially a giant fusion reactor? It's the power source for all life on Earth, and scientists have been striving for decades to replicate this process here on Earth. Today on Science Quest, we're diving into the thrilling world of fusion energy, a potential game changer that could solve our energy crisis and pave the way for a cleaner, more sustainable future. Unlike fission, which splits atoms, fusion combines them. Imagine two tiny hydrogen atoms, like shy kids on a playground. They're hesitant at first, but with enough heat and pressure, like a really enthusiastic playground supervisor, they get close enough that their cores fuse together, creating a helium atom. This fusion process releases a tremendous amount of energy, far more than fission or even the best fossil fuels. It's the same power that lights up our sun and other stars. But replicating the sun's power here on Earth isn't easy. We need to create conditions of extreme heat and pressure, millions of degrees Celsius, and immense pressure to force the hydrogen atoms to fuse. This requires incredibly sophisticated technology and massive amounts of energy just to get the reaction started. Think of it like trying to hold two magnets together with the same poles facing each other. It takes a lot of force. Despite the challenges, recent years have seen remarkable progress in fusion research. The International Thermonuclear Experimental Reactor ITER project, a global collaboration involving 35 nations, is building the world's largest tokamak, a donut-shaped device that uses magnetic fields to confine and heat plasma to fusion temperatures. ITER aims to demonstrate the scientific and technological feasibility of fusion energy. It's a monumental undertaking, a testament to human ingenuity and a collective desire for a clean energy future. Successful fusion energy generation could revolutionize many aspects of our lives. Imagine a world free from the constraints of fossil fuels, a world with abundant clean energy for transportation, industry, and even space exploration. Fusion power could potentially fuel spacecraft for interplanetary travel, dramatically reducing travel times and opening up new frontiers in space exploration. While solar, wind, and other renewable energy sources are crucial for a sustainable future, they have their limitations. Fusion energy offers a potentially limitless, clean, and safe source of energy, free from greenhouse gas emissions and nuclear waste concerns, and is a powerful alternative complementing other renewables to build a resilient energy grid. Of course, challenges remain. Fusion energy is still in its developmental stages, and commercial-scale fusion power plants are still some years away. But the recent breakthroughs and continued advancements inspire hope. The quest for fusion energy is a long-term project, a marathon, not a sprint, but the potential rewards are immense. The quest for fusion energy represents humanity's relentless pursuit of scientific advancement and its commitment to building a sustainable future. From harnessing the power of the stars to solving our energy crisis, the journey is filled with challenges, but the potential impact on our world is truly transformative. Next time on Science Quest, we'll explore the mind-bending world of quantum computing and its potential to revolutionize medicine, materials science, and artificial intelligence. Stay tuned. Science Quest, Unlocking the Universe Episode 1, The CRISPR Revolution and Gene Editing. Did you know that we're on the verge of writing the code of life itself? Forget science fiction, gene editing is here, and it's revolutionizing medicine, agriculture, and even our understanding of evolution. Welcome to Science Quest, where we explore the cutting edge of scientific discovery. Today, we're diving deep into the world of CRISPR, the revolutionary gene editing technology that's making headlines and changing the world. At its core, CRISPR-Cas9 is like a highly advanced pair of molecular scissors. Imagine having the ability to precisely cut and paste DNA, the instruction manual for life. That's exactly what CRISPR allows us to do. This incredible technology is derived from a natural defense mechanism found in bacteria's system they use to fight off viruses. Scientists harnessed this natural process and adapted it for gene editing. But how does it work? The CRISPR-Cas9 system consists of two main components, the guide RNA molecule and the Cas9 enzyme. The guide RNA is like a GPS, precisely targeting a specific DNA sequence within a cell. Once the target is located, the Cas9 enzyme acts like molecular scissors, cutting the DNA at the precise spot. Scientists can then introduce a new gene or modify the existing one, essentially correcting genetic errors or introducing new traits. The implications are staggering. Imagine a world where genetic diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease are a thing of the past. CRISPR offers the potential to cure these inherited disorders by directly correcting the faulty genes responsible. Clinical trials are already underway, showing promising results in treating various conditions. Beyond human health, CRISPR is transforming agriculture. Scientists are using it to develop crops that are more resistant to pests, diseases, and harsh environmental conditions. This could be crucial in addressing global food security challenges and ensuring sustainable agriculture practices. However, the CRISPR revolution is not without its ethical considerations. The power to alter the human genome raises profound questions about germline editing, modifying genes that can be passed down to future generations. This opens the door to potential unintended consequences and sparks debates about design of babies and eugenics. Responsible development and regulation are crucial. Scientists, ethicists, and policymakers are collaborating to establish guidelines and ensure that this powerful technology is used ethically and responsibly. Robust discussions and public engagement are essential to navigate the ethical complexities of gene editing. The future of CRISPR looks incredibly bright. Scientists are constantly refining the technology, improving its precision, and expanding its applications. We're seeing the development of base editing, a more precise technique that allows for single-letter changes in the DNA sequence, and primer editing, an even more sophisticated approach. The potential for advancements in this field is truly limitless. Imagine a future where genetic diseases are eradicated, Food security is guaranteed, and environmental challenges are mitigated through the power of gene editing. That future is closer than we think thanks to the incredible advancements in CRISPR technology. That's all the time we have for today's episode of Science Quest. Next time, we'll be exploring the mind-bending world of quantum computing and get ready to enter the realm of qubits and superposition. Until then, keep questioning, keep exploring, and keep seeking answers. This has been Science Quest. Science Quest, Unlocking the Universe Episode 1, CRISPR the Gene Editing Revolution. Ever wish you could edit your DNA like a word processor, correcting typos and upgrading your software? Well, thanks to CRISPR-Cas9, that science fiction is rapidly becoming science fact. This revolutionary gene editing technology is rewriting the rulebook of biology, offering potential cures for genetic diseases, boosting agricultural yields, and even reshaping our understanding of life itself. Prepare for a deep dive into the world of CRISPR, the gene editing revolution. Let's start with the basics. Our DNA, the blueprint of life, is a long, twisted ladder, the famous double helix made of 4 chemical bases: adenine A, thymine T, guanine G, and cytosine C. These bases form the code that determines everything from our eye color to our susceptibility to certain diseases. Genes are specific segments of this DNA code that instruct our cells to build proteins, the workhorses of our bodies. Now imagine a tiny pair of molecular scissors. That's essentially what CRISPR-Cas9 is. CRISPR stands for clustered, regularly interspaced short palindromic repeats, a naturally occurring defense mechanism in bacteria. Cas9 is the enzyme that acts as those molecular scissors. Scientists cleverly repurpose this system, using a guide RNA molecule to direct the Cas9 enzyme to a specific location within the DNA. Think of the guide RNA as a GPS precisely navigating the Cas9 scissors to the targeted gene. Once the Cas9 enzyme reaches the target location, it makes a precise cut in the DNA. This cut triggers the cell's natural repair mechanisms, allowing scientists to either disable a faulty gene or insert a new, corrected gene. This process holds immense potential for treating genetic diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease, conditions currently incurable. But CRISPR potential extends far beyond human health. Imagine a world with disease-resistant crops capable of thriving in harsh environments, minimizing the need for pesticides and maximizing food production. CRISPR is already being used to enhance the nutritional value of crops and improve livestock breeding. However, like any powerful tool, CRISPR comes with ethical considerations. The ability to alter the human germline, the DNA passed down through generations, raises significant ethical dilemmas. We need careful regulation and robust public discussion to ensure responsible development and application of this technology. What are your thoughts? Should we proceed with caution or embrace the potential benefits fully? Share your opinions on our social media channels. The future of CRISPR is incredibly bright. Scientists are constantly refining the technology, improving its accuracy and efficiency. Imagine personalized medicine, where treatments are tailored to an individual's unique genetic makeup. CRISPR could revolutionize cancer therapy, offering targeted treatments that minimize side effects. Further research could even lead to the development of gene drives, tools to modify entire populations of organisms, potentially eradicating invasive species or controlling the spread of diseases. But before we get carried away with visions of a perfect future, let's remember the importance of ethical considerations and responsible scientific practices. We need to ensure that CRISPR potential benefits are accessible to all and that its risks are carefully managed. Today, we explored the groundbreaking technology of CRISPR-Cas9, a revolutionary gene editing tool with the potential to reshape medicine, agriculture, and our understanding of life itself. From correcting genetic defects to creating disease-resistant crops, CRISPR possibilities seem limitless. But remember, the ethical considerations and responsible development of this powerful tool are paramount. Next time on Science Quest, we'll journey into the fascinating world of quantum computing, a technology promising to solve problems beyond the reach of even the most powerful supercomputers today. Get ready to explore the bizarre and wonderful world of quantum mechanics.